Alright folks, welcome to our wrap-up of Module 11 and the third unit of the course. We have been talking about repeated measures and factorial ANOVAs. Specifically, factorial ANOVAs has been the focus of the last couple videos. We're going to wrap up that factorial ANOVA material by talking about some of those post-hoc tests in this context, and also what changes when you have three or more factors at the same time. Spoiler, it's a lot more complicated. But fortunately, we just kind of zip past it and just talk about some of the specifics. Then we'll summarize things and put a pin in the module. So, just like with our one-way ANOVAs, when we reject the null, we have an unsatisfying statement. We have concluded there's some kind of a difference in the populations we're interested in, but not what it is or even how many differences there are. And again, factorial ANOVAs can have far more comparisons than a one-way ANOVA would because of the multiple factors. So just like with a one-way ANOVA, we can conduct post-hoc tests. A lot of times these are called planned comparisons because with more complex designs, there are so many different post-hoc comparisons you can make that usually it's encouraged to designate your strategies in advance. Otherwise, it can turn into a situation where you're just kind of mining for significant results. Because again, remember that idea of alpha inflation. If you run more and more comparisons, you're going to get more and more significant results, but a lot of them will be type 1 errors. So we generally plan these comparisons in advance, but some of the forms they can take, we're not going to get too far into the weeds in terms of calculation. But if you had a significant interaction, you might want to compare specific cells to one another to see the nature of that interaction. You also can do what's called probing the interaction, which basically involves testing how strong the interaction is at different sort of places in the distribution. It gets beyond the scope of this class, but there's a variety of ways of doing that. And it involves looking at how the interaction changes at kind of different score levels. So again, you're going to be following up on that interaction by making these comparisons. With a main effect, if you only had two conditions on that factor, well, you already know that they significantly differ. But if you have three or more on that factor, you need to follow up on it the same way you would with a one-way ANOVA. So which comparisons you make depends on what you're expecting to see as a researcher. And then sometimes we make some unplanned comparisons based on patterns of means we see in the data set that we did not expect. Conducting the planned comparison, again, there's lots of ways of doing this, but the most common way is to take the groups you're interested in comparing and either conduct a one-way ANOVA or perhaps a t-test if there's only two things, two cells that you're comparing, and again, basically conduct a follow-up hypothesis test. This would be a big pain to do by hand, of course, but to software, it takes, another second or two, and it'll give you those results. So I mentioned that the book calls factorial ANOVAs two-factor ANOVAs because they only really teach it in terms of having two different factors. In the real world, sometimes we run research that has three or more factors or independent variables. Parsimony is very, very important. We keep it as simple as possible, but sometimes we have legitimate reasons for looking at three or more factors, IVs or quasi-IVs at once. So here are some things that change. First, our notation changes. Remember the little equation that we make to designate our design? A three-factor or 3IV design would have three numbers in its notation. So a three-factor design might be described as a 2 by 3 by 2. What that's telling you is that we have three factors or IVs. Two of them have two conditions, the other has three. And again, notice it doesn't matter what order those numbers come in. You could call it a 3 by 2 by 2, a 2 by 2 by 3. All that matters is that it's telling you have three different factors. Two of them have two different conditions, and the third has three. And again, you multiply and you find 12 cells. Now the number of interactions increase. We have to test for every possible interaction between two factors, but also the possible three-way interaction. What the heck's a three-way interaction? The two ways were hard enough. Let's take our caffeine and temperature example that we talked about in the last module, or excuse me, in the last video. And let's say that we also just measured people's gender, and we found that that relationship, the interaction between caffeine and temperature, was altered based on whether you were male or female. It was different for men and women. So we might find, for example, that for men, the pattern looks like this. This is the pattern of data we already saw. Caffeine consumption hurts performance in a hot room, helps it in a normal temperature room. But let's say for women, we found this pattern. This is exactly the opposite kind of set of effects. Here, caffeine consumption is positive in a hot room and detrimental in a normal temperature room. We see an exact reversal here. Caffeine and temperature still interact, but that interaction is qualified, it's altered, it depends on the gender of the participant. completely different interaction patterns for men and women. That's about the simplest example of what a three-way interaction might look like. If three-way interactions or higher are tricky, my old methods professor used to love to say they do surveys. Grad students and faculty oftentimes have trouble interpreting three-way interactions, let alone four-way or more complicated. So if this stuff is kind of mind-bending or hard to get your head around, you are in good company. People that do this for a living can struggle with this sort of thing. But again, the idea here is that our two-way interaction between caffeine and temperature is altered by that third factor, which is the gender of the participant. So notice the number of tests accelerates very rapidly, and the number of main effects accelerates a little more slowly. You wind up with a lot more tests. So for a regular old two-factor design, you got a main effect for each of your two factors. You have that possible two-way interaction. Add a third factor into the mix. Now you have three main effects. You have three possible two-way interactions. Every single pair of factors, whether they interact with each other, and then you have that possible three-way interaction. And again, all of them could be significant or not. Let's say you add a fourth one into the mix. Oh, it's going to be painful. 4 different possible main effects. 1, 2, 3, 4, 5, 6 different two-way interactions that would need to be tested. 4 different three-way interactions, and a whopper of a 4-way interaction would have to be tested as well. That's a lot of different hypothesis tests. That's a lot of different F-scores. Again, a computer can do it without too much trouble. Most of the problem is that alpha inflation risk. You get more type 1 errors, and also just interpreting it. It gets very mind-bendy, very fast around 4 independent variables. And 5 IVs, you really don't want to know. I've seen exactly one study, I think, that's actually been run in a lab that I was in that had 5 factors at the same time, and it was really, really, really complicated. Parsimony is important. We use the simplest design we can. Usually people don't go above 3 factors. That's usually about the max. Once in a while you'll see a little more complicated one. All right, so that's the end of our actual content. Let's wrap up the module and the unit by summarizing things. First, repeated measures ANOVAs. Again, when we use a within participants design or a within groups design, we use the ANOVA, we alter it to accommodate the change specifically by eliminating individual differences. So in the between groups variance, the numerator, it's already missing, it's already gone, it's not a factor because it's the same people in each group. And in the within participants or within groups variance, it can be eliminated mathematically through some calculations that we walked through. That denominator variance is now just called error variance because that's the only thing that makes it up. The biggest advantage of our within participants or repeated measures designs is they are more powerful. The biggest disadvantage is you get a much bigger risk of those order effects. And this disadvantage is the reason why a lot of researchers shy away from this. You mostly see it in longitudinal designs when you're testing people with long periods of time between individual measurements. The factorial ANOVA, again, parsimony is important. You don't throw stuff in there just to throw it in there, but a lot of studies will have compelling reasons to use multiple factors, IVs or quasi-IVs. You get more information with a minimal increase in expenditure, but also you get to see if existing relationships or hypothesized relationships are altered by those second factors. A lot of times we run these tests because we're looking for interactions. Maybe we take, again, an established effect, an established IV-DV relationship, we add in a second one and we see if that qualifies or alters it. If they interact, that tells us something very important about that original effect. Your hypotheses may deal specifically with interactions between variables, especially in that kind of context. So definitely be familiar because we haven't gotten into calculation or anything like that. There are not too many points have been made. So be familiar with the ideas of main effects and interactions. Be able to define them, be able to identify them. If I give you a list of cell means again, you should be able to pick out which main effects seem likely, which interactions seem likely. And we practiced that a little bit. We're going to have an example problem on that in our practice quiz as well. Usually most of those possible main effects and interactions won't be significant, but we look at all of them. And this is the point when we let computers take over. We don't worry about doing it by hand. The key thing there is knowing what this stuff means and how to interpret a set of results. So that is the end of the module and the unit. Next up, you are going to want to go through your practice quiz, mainly about main effects and interactions. You're going to want to do your exam review, and you are going to want to complete your assigned homework. So the next thing from here on out will be taking that third exam. You are very close to the end of the course at this semester, so keep your head down, power through. You're almost there. See you on the other side of exam